Opsonization. Opsonization, the way we've discussed it so far, can do one of two things. Again, it makes it more likely to be ingested by bacteria, so it increases the rate and likelihood of phagocytosis. And it can also activate complement. Okay. So it can trigger that complement C3 being split, and then we could have one of three things happening, which we look at with the next step here. So when it does activate complement, when it does activate complement, do you guys remember the three things that can occur?
Opsonization.
So opsonization, giving rise to this,
Inflammation.
the inflammatory process,
And the membrane attack.
and then finally the killing part, direct killing part of complement would be the lysis. Okay, the insertion of that membrane attack complex. This is just kind of a summary picture of how one antigen can cause a plasma cells to make antibodies as well as activate the T killer cell. So it doesn't have to go one way or the other. Both of those processes can be utilized. Okay, before we leave antibodies and look at passive immunity, I threw in just a few review questions. Get you guys comfortable so you don't just go, stop breathing when you see a slide that talks about blood antibodies. So, lab type question or lecture type question, identify the blood type shown below. AB negative, excellent. So an individual has antibodies A and antibodies D in his blood. This is like some of the ones I wrote up on the board. So if he has antibodies A, he has antigen B. If he has antibodies D, then he does not have the D antigen, so B negative. Okay. So Ken has been given a transfusion of AB positive blood and develops a transfusion reaction. Which of the following blood types does Ken probably demonstrate?
Any of them.
So his blood type is not AB positive. He was given AB positive blood.
E positive is everything, so it could be anything.
So if he has A negative blood, what antibodies would he have?
D.
He would have B antibodies, and he might have D. Would that cause a transfusion reaction?
Yes.
Yes. Okay, just the B antibodies alone would cause a transfusion reaction. If he has B negative blood, the same thing. He would have A antibodies, and he might have D antibodies. So the A antibodies alone would cause a transfusion reaction. If he also had D antibodies, that would just be even worse and occur faster. If he had O negative blood,
Yes, and B
that's
antibodies.
A and B antibodies, and maybe D. So now that would be the worst case, right? Because he could have antibodies against A and antibodies against B and antibodies against D, and those are all the antigens that are on the donated transfused blood. All right, now he has AB blood. So he's getting AB blood. If he has AB blood, is there any antibodies between the A and the B part? No. But again, he could have the D antibodies. And so we don't want to give him AB positive if he already has, if his blood type is negative. So the answer is E. All of these could have antibodies present against one or more of his antigens. Again, don't memorize those answers, okay? Follow the steps by which we got the answers. So what blood type should we give him?
O negative.
O negative. Okay? Because this is the one blood type that's the universal donor. We don't know his blood type. So we want to give him something that has nothing on the red blood cells that could result in a transfusion reaction. Does that make sense? We have no idea what his blood type is. Okay. So when we go through this process that I drew on the board earlier with the activation of the B lymphocytes, remember what had to happen first? The T helper cell had to be activated, and then the B cell could become responsive. Those don't have to happen at exactly the same time, but both events have to occur. That process, remember when we talked about the mother, how many days we had to, within which we had to give her the RhoGAM shot? 3 days, 72 hours? Okay, so there's that early span that we can stop that process. But it still takes a while to complete it. And so it takes 7 to 10 days for the primary immune response. That means for the T helper cell and the B lymphocyte that's never been activated before to become activated against that particular antigen. And those numbers of antibodies, there aren't very many active B lymphocytes that are formed. Some of them remain as memory cells, but the others don't make very many antibodies. So they only last for about a period of 4 or 5 days. You can still get sick with this primary immune response. It took so long for that to reach the peak, that's almost a little over 2 weeks to reach the peak of antibodies, that you've got a good time to get sick, and your secondary immune response, the complement and the fever and the inflammatory response, may not be sufficient to keep the bacteria or the virus population in control. All right, so you could get sick. Now the second time around, it's hours to a day or so. Those memory cells are present, they multiply, we have a very rapid response, a higher level of antibodies in the blood, and they last for a much longer time. This will keep you from getting sick from that particular antigen. Okay? So that's the primary versus secondary response. This is just showing another primary response to a different antigen.
So the higher is a secondary response
Yes.
to, okay.
I'm not sure if it's very clear here where the exposure is, but here and then here is the exposure to each antigen. So 7 to 10 days, barely, that's 28, barely a couple of days there. But the process would start within a couple of hours. All right, let's finish up here. I might actually finish early and you guys can ask some questions anyway. So types of immunity. This is very basic, so don't let it confuse you, all right? You just have to think about what the criteria are. And we're talking about humoral immunity here, okay? We're talking about the B lymphocyte. So active means that your body has the B lymphocytes that are the source of the antibodies, and retains their memory cell components. That's active. Passive means all you get are the antibodies. Once the antibodies are older and are broken down, you no longer have antibodies. Okay? That's the difference between active and passive. Natural means there's no injection involved. No spray, no sugar cube. It is a natural process of introducing either the antibodies or the antigen. All right?
So no chemical stimulation?
Right. No external. It's a natural exposure. I mean, the virus could be external, but you got it in a natural manner. All right, so types of immunity. So we're going to combine those two terms, either passive or active, either natural or acquired. Okay? So here we have the fetus in the amniotic sac, getting antibodies crossing the placenta. Remember the IgG antibodies would be crossing the placenta and getting to the infant, and cross the amniotic membrane as well for that matter. And we have a mother breastfeeding and IgA antibodies in the breast milk. Any discussion of the baby making its own antibodies here? No. But no injection, all right? So this is a natural process. A natural means by which the infant is obtaining the mother's antibodies. Passive, because the baby's not making them. The baby hasn't been introduced to the antigen. It's receiving the mother's antibodies, and the mother retains the B lymphocytes and can continue to make them. Once the antibodies are old, broken down by passing through the liver, they're gone. The baby has to continue to be fed breast milk if it's going to maintain its level. Once it's weaned, it's not going to keep those A antibodies in its blood for more than a few months. Okay? So passive, but a natural passive process. So we've talked about these two aspects already, the RhoGAM shot. So the gamma there stands for gamma globulins, which is like an immunoglobulin. So those were raised in a goat or a horse, into which D antigens were introduced. They're still passive because we're not giving the antigen to the person. We're not giving the Rh factor to the person. We're not giving the snake toxin to the person. We're giving the individual antibodies that were developed against that in a different organism. Hence, passive. The individual getting the antivenom serum or the RhoGAM shot is not making those antibodies. They're being given them by injection. Okay. Does that make it, you understand the difference between that passive and this passive? Okay, an artificial environment. So acquired. Then we have active natural immunity. You get sick. So the virus crosses your mucous membranes and you get chicken pox or you get the flu. Now the T helper cells and the B lymphocytes go through the pathway we just described earlier, and you retain B memory cells. So the second time you get exposed to the chickenpox virus, those viral particles and viruses are destroyed before they have a chance to multiply and result in illness. So it's active. You're making your own antibodies and you keep the B lymphocytes afterwards, and it's a natural process. We didn't expose you. Now, okay, so chickenpox parties may not necessarily be natural, but the mechanism by which those kids develop their antibodies would be, okay? They're getting sick. They're not getting the antibodies from the sick child. They're getting the virus, and then they're getting sick and developing subsequent antibodies. Now, my oldest son got the chickenpox, very mild. We had been at a ball game, spring training in Arizona, and we were supposed to go, my husband and I were supposed to get back in time to go to Phantom of the Opera, and he had arranged for visits behind the stage afterwards and, you know, all that kind of reception stuff. And we stopped off at my sister-in-law's in Fresno, and I was changing a diaper kind of in the dark. And I noticed he'd had a fever at the ballpark and in the car on the way back, driving back across the border. And when I changed his diaper, I noticed 3 little red spots on his bottom. And that was really all he had of chickenpox. I don't know how many kids he infected at the ballpark. Because with the fever, he was infectious. So we stayed home, didn't go to Phantom of the Opera. He never had a shot. Now my twins, by the time they were born, 5 years later, they got a vaccine against chickenpox. Okay? So they never had to get it. They will be probably not get shingles or be at risk for shingles when they get older. So that's, you know, everybody thinks getting chickenpox is a rite of passage, but then shingles is a very serious disease and later on in life, as far as the pain level goes. Having gotten it on Christmas Eve myself. Okay.
That is not real.
No.
It's a mean picture.
Very
I don't know what they did to the baby to get it to get that face, but all right, so active acquired. So this is when you're given a vaccine, which is a weakened virus. Now we talked about viruses, T cells, you know, T killer cells being developed, but these are the antibodies not destroying the viral infected cells, but the virus itself. All right, so they were desensitized to the virus itself. And so we don't want to get a person sick. We don't want to give them polio to get the antibodies against the polio virus. And so an attenuated virus. It means one that's still alive but has been weakened. It's not going to be able to cause the disease. It's given, so it still has immunogenic factors. It can still activate T helper cells and B cells. And now you will have B memory cells. So the next time you're exposed to that virus, you'll have that secondary immune response, all right? The reason you get sick with the primary immune response is because of the attenuation of the virus. So you can get the vaccine, you can get the immunity without actually getting the disease. We're done. Something about this room, I think we finished early.
We're pressed for time.
So the hypersensitivities that are at the end of this, other than what we said about IgE, I'm not going to word that. Yeah.
So when we're talking about the B lymphocytes, they incorporate